Hi, I'm Aaron from DataClassroom, and today I'm going to show you how to share a data set with your class, and then I'm going to show you how to work through a data set from the student perspective. And the activity data set that I'm going to be using as an example is Potato Osmosis. And I think this is a good one if content-wise you're trying to cover cell transport, osmosis diffusion, maybe you're wanting to model how water moves across a cell membrane by diffusion using a real data set. This would be a good example to do that. And I will say this is one of the most classic of all classic biology or life science experiments. And I am a fan of this activity. I did it for years with my own students. Um, okay, so first thing, let's go ahead and locate this data set from the resource library. If I go up to the Browse tab up there, you can see the red circle where I'm clicking. I'm going to come down to Resource Library right there like that and click on resource library. I already started searching for this, but just to show you what it would look like, this is the resource library when you come in, and I'm going to search osmosis up here in this field. So I type in osmosis and I get this one. You can open the lesson plan, but I'm just going to go straight to the data set and we're going to start working with this. So I open the data set. And here we are. This is the data set view. Within a data set, we have a table view, we have graph view, and then we have any attachments. In this case, we've attached an assignment. Now, a lot of our older data sets will not have this because this is a relatively new feature in DataClassroom, but this one is going to use the assignment tab quite a bit. So I'll show you how to do that when we are working through the assignment as a student. Right now, I'm going to show you how to share this data set with your class. So your students can find this one in the resource library or in the data set list, but say you make a change to it. You make a custom version, you add a question, you take away a question, you add some more data. Maybe you're using your own class data, but any of those things, you make your own version, you want to share it with your students. So all you have to do to make your own version is make a change that you want, and then you go up to save as, right? And so, we'll call this potato osmosis lab data and we'll call it the Mr. Reedy version. Okay. And click save. And you'll notice the DataClassroom branding goes away. That's reflective of this is now your own private copy. It's no longer the copy that's in the resource library. This is your own. And you can find this if you go up to browse and data sets, you'll be able to see all data sets, not just ones in the resource library, but yours and your students as well, if you've integrated with a learning management system. Now, if I want to share this data set with my students, I go over here to this hamburger menu in the upper left on the white bar, and I get this slide out, and the third option down is share right there. And I can share with the class. And when I click this drop down, I'm going to see all of the classes that I've rostered with using Schoology or any other LMS, Google Classroom, Clever. I can go ahead and I can pick the class I want to share with. So I'm picking a class called Teacher Workshop 2018. You can pick your own classes from the list, and you click share a copy, and that will make a copy and share it with your students. So they'll have it appear in their list of data sets. Um, Another way you can share is you can click this anyone with a link can view. And so maybe you want to put this on a class web page or something like that. I copy that data set and I can just post that link wherever I want my students to be able to find it. This is very similar to how Google Drive works. Notice you can also click anyone with a link can edit. And that when you share that link, when that's been checked, that button's been pushed, then anyone who finds that data set can actually make changes to it. I'm going to turn that off for now. So that's how you share a data set with your students. Now, from this point forward in the demonstration here, I'm going to say that I am a student. And the reason I can do this is the student view and the teacher view are not really different from each other. There's certain things like share with a class that students just won't have available. So they will not see that option for sharing a data set. But everything else is largely the same between student view and teacher view. So this makes it simple for you as a teacher to see what your students are going to see. So now I'm going to pretend I am a student and work through this. So I open this up to the data table view, and I say, okay, what do I need to do here? I'm going to go over to the assignment tab. So I'm up here at the top with the little paper clip where it says assignment, and I'm going to go to the assignment. And this is opening up a view of this Google Doc, which has been embedded in here. And I start reading through this, and I read the background, and I look at the illustrations. And this is giving me the background that I need to answer the questions in this activity. There's even a video if I want to get a more accurate picture of what this procedure was that generated this data set. And I go down to activity, and it's going to tell me, I got to start out by making a graph. I'm going to choose the scatter plot icon, show sucrose concentration on X, mass change on Y. Um, if I want to see what the instructions are to actually do some of these things in DataClassroom. Now, these are very basic actions within DataClassroom. So if your students have used it before, they will probably know how to make a graph. But if this is their first time, they can actually click the link in this document, and it will open up the instructions for making a graph. So it shows that I need to click the graph tab, shows me how to add variables, even a short little YouTube video. I think this one's just a couple minutes long if they want that. So those instructions are there if they need them. But I'm going to go back to my data set. And I got away from the assignment. So I'm going to go back to the assignment, and I'm going to go down to that item number 1 in the activity where it's asking me to make a graph, add descriptive statistics, and paste it here. So Potato Osmosis Lab, remember again, this is where I've exposed little pieces of potato. I've submerged them in either a salt or a sucrose solution for some period of time, typically 12 or 24 hours. And then I've recorded the change in mass that occurred while that piece of potato was soaking there. Now, just a reminder for this lab: any change in mass here is due to osmosis, so water moving across the cell membrane, either entering those plant cells or exiting those plant cells. And that's the background of the lab. But let's go and make that graph. And so I can't go directly into this doc and start working on it. But if I'm a student, I can go over here and I can click edit, and it's telling me what's going to happen. I'm going to click yes, edit it. But what it's doing is it's creating a new Google Doc in the DataClassroom Google Drive. And as a student, I am the only person who has permission to edit this doc. So it is my copy to work with. And so here you see it's opening up this Google Doc, and you can see the file name indicates that this is something that resides in this DataClassroom Google Drive, but I have permission to edit it. So I can go down to my question number 1, and you can see where it says paste graph here. So I'm going to go and make that graph, and then we'll take it and we'll put it in here. So I'm going to go back to DataClassroom, and I'm going to go to the graph tab. And now to go ahead and make this graph, I'm going to just click the variables that I'm interested in. So mass change percentage. And then I'm going to go get sucrose concentration polarity and click show. And now I've got this graph. Now, this is what's called a jittered dot plot. So jitter means the horizontal spread of the points. And I can control the jitter over here with this plus minus. So more jitter down to no jitter. And so this is where all the dots are stacked on top of one another. I'll give it 1 unit of jitter. I can see sucrose concentration is being treated as 6 different categories rather than a numeric variable. And we'll change it a little later on in the activity to a number. And so you can see what different options that will give us. But for each treatment, 0, 0.2, 0.4, 0.6, 0.8, 1.0, these are each of my 6 different sucrose solutions. And I can see all the points for that group. Now, if I want to add descriptive statistics, I go over here, click this checkbox at the control panel at the right, and it's displaying a median with a box and whiskers. Maybe I want to see a mean, so I can go and click that, and get a mean based. Now it's showing me a mean and standard deviation for each. Maybe I want to do, 2 standard errors of the mean. Now it's showing that. So you can play around with what you want those error bars to be showing around the mean. And it's also, it's calculated that I'm going to move me over here so we can see this table at the right. But you can see for each treatment group, it's displaying the mean, standard deviation, standard error, and so forth in this table here at the right. Now, if I want to put this into my answer sheet that I'm working on, I can click the camera in the upper right here, and it's going to copy this image. I'm going to click copy and done. And now it's on my clipboard. So if I go to my answer sheet that I'm working on, I can paste in my graph. And, maybe I want to make it a little bit smaller. I'm just doing, an aesthetic change here. I'm going to get rid of the sentence that says to paste the graph here because I don't really need it anymore. And so now I've got my graph pasted in there. Now I can continue in this fashion to edit this doc and use it as my answer sheet. So what are the independent and dependent variables in this experiment? So I'll say the IV is sucrose concentration, and the DV is change in mass. And I can enter those in, and that's in my answer sheet. I'm going to work through this until we have to go back and make another graph, so I can continue to work in here. How does mass change with sucrose concentration? Here we can say as sucrose concentration increases, mass change decreases. And I will provide you with an answer sheet here. For any of the answer sheets for DataClassroom, you can send us an email at info@dataclassroom.com and request them. We don't put those publicly on the web to minimize the number of those that are floating around with students. I'll also include it in this materials package as well. So which substance moves across the cellular membrane in this activity? That would be water. That's what is responsible for the change in mass. And then what is the specific name of the movement in terms of this substance? That is osmosis. And that's when water is moving across the cell membrane. Now for question 5, we have to make another graph. And what it's asking us to do is change the variable. And again, remember this link here, where change the variable in blue, that's a hyperlink that will show us how to change the variable. I'm just going to demo that for you. And we want to add a regression line of best fit once we've changed this to a numeric variable. So let's go back and do that. So here I am. I'm going to go up to the column header for sucrose concentration molarity and see where it says categorical. I'm going to click that, get this drop down. I'm going to change it to numeric, and it pulls it off the plot. So now all we see displayed is mass change percentage, but I'm going to add it back by clicking show. And now we have it added back as a numeric variable. And what you'll notice, that jitter is gone because we can no longer move points horizontally for display purposes, because now the horizontal position of those, or their position along X, has a numeric value, because these are now being considered numbers. But you can see I have new options over here on the control panel at the right. I can see things like regression line, which if I add regression line, I can see that across there. And so this is showing you the overall trend. And with this lab, a lot of times we're concerned with where does this intercept the x-axis, is because this is a sort of a approximation of what the solute concentration is within the cell. And this would be the solute concentration where the net water gain or water loss was 0, right, where there's no change in mass. And you can see over here at the right, the x-intercept is displayed here. So it's 0.2546 if you wanted your students to find that in the course of this activity. Um, but this helps display this. And again, if I want to use this in my uh, answer sheet, I'm going to copy that image again, and I'm going to go back to my answer sheet where I was working, and maybe I want to paste that in there. So now I've pasted that in with the line of best fit. Um, so my best estimate for the natural solute concentration inside the potato cell, again, what I'm doing is taking the point where this crossed the x-axis, and I believe that was 0.2546. Um, that is probably, and that's in molarity. That is probably the a pretty good estimate for the natural solute concentration inside there. And of course, my data is evidence for that estimate, because that is the point where the line of best fit crosses the x-axis. And which solution is closest to being isotonic with respect to a potato cell? Um, that is the zero point 0.2 sucrose concentration. It's very close to that 0.2546. Um, that is isotonic. And then which solutions were hypertonic, which were hypotonic? The hypotonic solution was the zero point 0.0, the pure water. And that we know that because the cell gained mass, it gained water. And all of the others, the 0.4, the 0.6, the 0.8, the 1.0, those were hypertonic and caused the cells to lose water. And then we have the challenge question, which is, using the principles illustrated with these data, explain why you can't drink seawater when lost at sea. Well, if you drink seawater, which has a high salt concentration, you're raising the solute concentration of your blood and the fluid outside of your cells. So relative to the outside, you have more water, higher water concentration inside the cell, and it will cause water to flow out to that area of lower water concentration or higher solute concentration, and those you will become dehydrated at a cellular level, which is why you cannot drink seawater when lost at sea. Um, so that is pretty much the walkthrough. I'll go back to the data set here. That is pretty much the walkthrough. I want to just show you, one more graph you might be interested in making. I'll take regression line off here. One thing you might want to do is have your students look at their data relative to all the other lab groups' data, and we can do that because we have this variable called lab group name. And if I add this to the plot with show, you can see it gets queued up at the control panel at the right, but it is grayed out because it's not showing. And I don't want to show it on X. I don't want to show it on Y. What I want to do is make it what we call the Z variable. And I add it on Z. And now you can see it's color coded by lab group. And if I want to do something like now, like, connect the dots, I can do that, and it's doing those by the data series for each lab group. And what you see here is kind of what I call the spaghetti noodle effect, where all of these are kind of intertwined and stacked on top of each other, um, indicating that one group doesn't stand out as having really high readings or really low readings, indicating that basically you're seeing the same thing no matter which lab group you looked at the data for. And so that is pretty much it. That is the Potato Osmosis lab data set and activity. Again, if you want to customize it, you can change anything about it. You can change the attachment, that Google Doc that we worked from. You can make your own version and share that with your students. You can change the data. You can do whatever you want. And then when you share it with your students, they can actually use that document, make a copy, and put their answers directly in there. If you have any questions about how to work with a data set in DataClassroom, how to implement it in your class, don't hesitate to reach out. So with that, I'll say thanks for watching.